massive biological energy conversion. And if you just look at the big picture equation, it seems almost, well, deceptively simple.
Right.
You take glucose, C-H, oxidize it step by step with 6 oxygen molecules, 6 euros, and what you get out is 6 CO, 6 H, and the big prize, somewhere around 30 to 32 ATP molecules.
And that little bit of wiggle room, that 30 to 32 range,
Yeah.
that's a key clue right away, isn't it? That we're not just dealing with some simple battery here.
Exactly. It tells you there's variability, there are nuances.
So we're definitely getting into some dense territory. Our mission today, for you listening, is to go way beyond just the textbook summary. We want to break down exactly how we reach that, that variable ATP target. We'll focus hard on the 3 link stages, the really key regulatory points, and maybe most surprisingly, the medical side of things. Like, why does a common diabetes drug or even a deadly poison target this exact machinery?
Yeah, understanding that energy flow is absolutely essential. It explains why life works and, frankly, why it fails so catastrophically when something breaks.
Yeah.
So we've got a pretty clear itinerary. We'll follow the carbon atoms. First stop, glycolysis. That's the anaerobic kickstart out in the cytosol.
Okay.
Second, we move into the mitochondria for pyruvate oxidation and the citric acid cycle.
Right, the Krebs cycle.
And finally, the grand finale, oxidative phosphorylation on the inner mitochondrial membrane. That's where the big ATP payout happens. But we also need to spend some real time on the backup plan, fermentation. That's what happens when oxygen just isn't there.
Okay, so let's start outside mitochondria, right there in the cell fluid, the cytosol. Glycolysis.
Mm.
The name itself, sugar splitting, tells you what it does. And it's ancient, right? Like the most fundamental pathway.
Absolutely. It's foundational. Every cell does it, from bacteria to us. And crucially, it doesn't need oxygen. It's anaerobic.
So the goal here is what? Just a quick bit of energy.
Well, 2 things, really. First, yes, capture a small, fast burst of energy. And second, prep that glucose remnant, which is now pyruvate, for potentially, more processing inside the mitochondria later on.
Okay, and the net yield. It's not huge at this stage.
No, it's modest but vital. Per glucose molecule, you get a net of 2 ATP. That's made by substrate-level phosphorylation. We'll get into that. You also get 2 NADH, which are really valuable energy carriers. And, of course, the 2 pyruvate molecules, the leftover bits of the sugar. But, importantly, before you get any of that, you have to spend some energy first.
Right, the investment phase. You got to spend money to make money.
Exactly. Glycolysis has these 2 distinct phases. Phase 1, energy investment. Here, the cell actually uses up 2 ATP molecules. Think of it like tagging the glucose, making it unstable, ready to be split.
Okay, so where's the first ATP used?
Almost immediately. Step 1, an enzyme called hexokinase or glucokinase in the liver uses 1 ATP to stick a phosphate onto glucose, making glucose 6-phosphate.
Got it. But it's the second ATP investment that's the really critical one, isn't it? The point of no return.
That is absolutely the key regulatory point in glycolysis. It's step 3. The enzyme here is phosphofructokinase 1, or PFK1 for short. It takes fructose 6-phosphate, uses that second ATP, and makes fructose 1.6 bisphosphate. Once that second phosphate is on, that glucose molecule is committed. It will be split and processed further.
So PFK1 is the bottleneck, the rate-limiting step.
Without a doubt, it controls the pace of the entire pathway.
Let's dig into that control, because PFK1 isn't just, mindlessly working. It's sensing the cell's energy levels, right? Like a little metabolic accountant.
That's a great analogy. It uses allosteric regulation, meaning molecules bind to it away from the active site to turn it on or off. It's basically reading the cell's energy bank account.
So what signals does it respond to? What tells it to speed up or slow down?
Primarily the cell's energy charge and whether downstream pathways are backed up. So if the cell has lots of energy, high levels of ATP, PFK1 gets inhibited, slows down.
Makes sense. Bank's full.
Right. And interestingly, high levels of citrate also inhibit PFK1.
Okay, citrate, that's from the citric acid cycle, which happens inside the mitochondria. Why does that affect PFK1 out here in the cytosol?
That's a really important link. When the citric acid cycle inside the mitochondria gets backed up, maybe because the cell has plenty of fuel as acetyl-CoA and isn't using it fast enough, citrate starts to build up and gets transported out into the cytosol.
Ah, so it's like a signal from the mitochondria saying, hey, we're full down here, stop sending more fuel.
Precisely. It tells PFK1, hold off on processing more glucose. Conversely, if the cell is low on energy, signals like high AMP, that's adenosine monophosphate, a sign ATP is really depleted, strongly activate PFK1. And another activator is fructose 2-6-bisphosphate, or F2,5,6-BP. These signals can actually override the inhibition by ATP, saying we need energy now. It's quite sophisticated.
Very cool. Okay, so PFK1 commits the glucose. Then that fructose 1,6-bisphosphate gets split by aldolase into two 3-carbon molecules, DHAP and G3P. And these can be converted back and forth. Now we're into phase 2, the energy payoff. And everything happens twice from here on out, right? Because we split the sugar.
Exactly right. Now the cell starts getting its return on investment. The first big energy capture happens in step 6. Glyceraldehyde 3-phosphate dehydrogenase, quite a mouthful, oxidizes G3P. And this is where we generate those first two crucial energy carriers, 2 molecules of NADH.
And the ATP generation here, you mentioned it's substrate-level phosphorylation. That means it's direct, right? No mitochondria needed yet.
Correct. It's very direct. In step 7, and then again in step 10, oh wait, step 9 actually, apologies. A phosphate group that's attached to the substrate molecule with a high energy bond is literally just transferred directly onto an ADP molecule, making ATP.
Simple transfer.
Simple transfer. In step 7, it comes from 1,4-fucosylbisphosphoglycerate. In step 9, it comes from phosphenolpyruvate, or PP. Since this happens for each of the 3 carbon molecules derived from glucose, we generate a total of 4 ATP this way.
4 ATP generated, but we spent 2 earlier.
So the net gain is 2 ATP, plus those 2 pyruvates and the 2 NADH we made earlier.
And the enzyme doing that final step, step 9, pyruvate kinase, it's also regulated.
Yes, it is. And it shows this really neat mechanism called
You can do
feed-forward
it.
activation.
Let's work
like
today
PFK1,
to make
it's
tomorrow
inhibited by
a better
signs of high
day.
energy like ATP itself,
Okay,
and also the
you
amino
can
acid
do it.
alanine. But
Let's
it gets
work today
activated by
to make
fructose
tomorrow a better day.
phosphate, the product of that
Okay.
PFK1 step,
You
way
can
back
do it.
upstream.
Let's-
So PFK1 is basically telling pyruvate kinase, hey, I just committed a big batch of glucose, get ready, stuff is coming your way.
Exactly. It ensures the whole pathway stays coordinated and doesn't get jammed up. It keeps the pipeline clear when the input is high.
Okay, glycolysis done. We've split the sugar, got our 2 pyruvates, a small net gain of 2 ATP, and 2 NADH carriers loaded with energy. Now, if oxygen is available, and that's the big condition, pyruvate takes a trip, it moves from the cytosol into the mitochondrial matrix.
Correct. This is the transition point. And it's controlled by a real gatekeeper, this enormous enzyme system called the pyruvate dehydrogenase complex, or PDH.
PDH. And its job is to convert pyruvate into something the next cycle can use.
Yes. Pyruvate has 3 carbons. The next stage, the citric acid cycle, needs a 2-carbon molecule called acetyl-CoA to start. PDH makes that happen. It chops off 1 carbon from pyruvate as co-urose, attaches the remaining 2-carbon unit to coenzyme A, that's the CoA part, and makes acetyl-CoA. And in the process, it also generates another molecule of NADH. This whole reaction happens twice for every starting glucose molecule, remember, because we got 2 pyruvates from glycolysis.
You called PDH enormous, a complex. Why does just chopping off 1 carbon need such a big machine? You mentioned 3 enzymes and 5 cofactors.
It's because this step is absolutely critical and, importantly, irreversible under normal cell conditions. Once you make acetyl-CoA from pyruvate, those carbons are basically committed. They'll either be burned completely in the citric acid cycle or used to synthesize fats. There's no going back to glucose easily from acetyl-CoA.
So the complexity is for control and efficiency.
Exactly. The 3 enzymes work together, and those 5 cofactors, thymine pyrophosphate, TPP, lipomide, FAD, coenzyme A itself, and NAD, each has a specific job in passing the acetyl group along and handling the electrons.
Can you give us a quick sense of why a couple of those are needed, like TPP and lipomide?
Sure. TPP, which comes from vitamin B1, is essential for the first step, the actual decarboxylation, removing the CRO. It holds onto the 2-carbon acetyl group temporarily. Then lipomide acts like this long, flexible arm. It swings over, picks up the acetyl group, forming acetyl-CoA. It's like an internal delivery system within the complex.
Wow, okay. Like a little assembly line.
Pretty much. It prevents these high-energy intermediate molecules from just floating away. Then FAD and NAD are needed to reset the lipomide arm and the complex back to its starting state, ready for the next pyruvate. That's where the NADH is generated in this step. And this explains things like beriberi, the disease from severe thiamine B1 deficiency. If you don't have enough TPP, PDH stalls, and you basically starve the cell's main energy pathway.
That really highlights PDH as the crucial link, the choke point between the glycolysis outside and the big energy production inside the mitochondria.
Absolutely. If you stop PDH, you cut off the main fuel supply to the citric acid cycle.
So regulation must be tight here too.
Extremely tight. And again, it follows the energy logic. If the cell has plenty of energy, high levels of ATP, or lots of acetyl-CoA or NADH, the products of the reaction, these signal PDH to slow down. Inhibition.
And activation.
Activation happens when the cell needs energy. Signals like calcium ions, Ka, often a signal for muscle contraction or cell activity, and ADP, the low energy signal, turn PDH on.
Okay, so PDH has done its job. We now have acetyl-CoA inside the mitochondrial matrix, ready for the next stage, the citric acid cycle.
Yeah.
Also called the TCA cycle, or often the Krebs cycle, after Hans Krebs, who figured it all out.
That's right, Sir Hans Krebs, a monumental achievement.
And the main purpose here isn't really making ATP directly, is it? It's more about harvesting those energy carriers.
Precisely. The goal of the TCA cycle is to finish the job started by glycolysis and PDH, to completely oxidize those last two carbons from the acetyl-CoA, stripping away all their remaining high-energy electrons. We load those electrons onto more NADH and another carrier called FADH, and we release the final carbons as COA.
Okay, let's walk through the cycle briefly. Acetyl-CoA, with its 2 carbons, comes in and joins with a 4-carbon molecule already in the cycle, oxaloacetate.
Yep, that forms the 6-carbon molecule, citrate. That's step 1, catalyzed by citrate synthase. Very favorable reaction.
And then citrate goes through a series of, 8 steps?
8 steps in total, yes. The really key energy harvesting events are the 2 steps where we lose carbons as CoO and make NADH. Step 3 is critical. It's catalyzed by isocitrate dehydrogenase. Here, the first CoAs is released, and we generate the first NADH of the cycle proper.
The isocitrate dehydrogenase. That sounds important. Is that another major control point like PFK1 was for glycolysis?
It absolutely is. It's considered the main rate-limiting enzyme for the TCA cycle itself, and its regulation mirrors PFK1 almost perfectly.
How so?
It senses the cell's energy state. High energy signals lots of ATP or NADH, shut it down. Inhibition. Low energy signals ADP or calcium ions, again, turn it on. Activation. It controls the flow through the cycle.
Got it. Then the second carbon comes off as CoO in the next step, step 4.
Correct. Catalyzed by alpha-ketoglutarate dehydrogenase. This releases the second and final coeuromolecule derived from that initial acetyl-CoA, and it generates another NADH. So if you're tracking the carbons from the original glucose molecule, by this point, after 2 turns of the cycle per glucose, all 6 original carbons have now been released as SUNORO. The fuel is fully oxidized.
The carbon skeleton is gone, but the cycle needs to continue to regenerate that starting molecule, oxaloacetate.
Exactly. The remaining steps do that, but there is one more small, direct energy capture in the cycle.
Ah, right. A bit like the substrate-level phosphorylation in glycolysis.
Very similar concept. In step 5, an enzyme called succinyl-CoA synthetase breaks a high-energy bond in its substrate and uses that energy to make 1 molecule of GTP guanosine triphosphate.
GDP, is that the same as ATP?
Functionally, yes. GTP can be very easily converted into ATP by the cell. So it counts as 1 ATP equivalent produced directly within the cycle. It's the only one.
Okay. Then we get the other energy carrier, FADH. Where does that come in?
That happens in step 6. The enzyme is succinate dehydrogenase. It oxidizes succinate to fumarate, and the electrons are transferred not to NADO, but to FAD, creating FADH.
And succinate dehydrogenase is unique, location-wise.
It is. It's the only enzyme of the entire TCA cycle that isn't floating freely in the mitochondrial matrix. It's actually embedded directly within the inner mitochondrial membrane. In fact, it doubles as complex 2 of the electron transport chain we'll talk about next. It's perfectly positioned to hand off its electrons immediately.
Very efficient design. Okay, the cycle finishes up, regenerates oxaloacetate in step 8, and makes one last NADH in the process.
That's the loop. Ready for another molecule of acetyl-CoA to come in.
So let's tally the score for the TCA cycle. Remember, it runs twice for each glucose.
Right, so per glucose, the total haul from 2 turns of the TCA cycle is 6 molecules of NADH, 2 molecules of FADHers, 2 molecules of GTP or ATP equivalent, and the release of the final 4 CO molecules.
That's a lot of NADH and FADH. The real energy payout is clearly still waiting in those carriers.
Absolutely. All that captured electron energy is now poised for the final stage, where it gets converted into the bulk of the ATP.
All right, this is it, the main event, oxidative phosphorylation. This is where, 90% of the ATP from glucose is actually made?
Easily. We're moving from the mitochondrial matrix to the inner mitochondrial membrane itself. This folded membrane is packed with the protein complexes that run the show. And the whole underlying concept here is Peter Mitchell's chemiosmotic theory. Brilliant work, got him the Nobel in '78.
Chemiosmosis, so it's about linking chemical reactions to pumping ions.
Exactly. Mitchell figured out that the electron transport chain, the ETC, wasn't making ATP directly. Its job was to act as a proton pump. It uses the energy from those electrons carried by NADH and FADHers to pump protons, H ions, from the matrix out into the space between the inner and outer mitochondrial membranes, the intermembrane space,
Creating a gradient.
creating a powerful electrochemical gradient. That gradient is stored energy, what we call the proton motive force. Then another complex uses the energy of that gradient to actually make the ATP. It's a two-step process.
Okay, let's break down the ETC first. It's a series of complexes, right? Passing electrons down the line.
Yep, four main protein complexes plus some mobile carriers. Electrons get passed from one to the next in a series of redox reactions, moving to lower and lower energy levels.
Where does NADH drop off its electrons?
NADH interacts with complex I, also called NADH dehydrogenase. It's a huge complex. NADH gives up its two high-energy electrons, becoming NADO again. Those electrons move through complex I, passing through intermediates like FMN and iron-sulfur clusters. As they move, the energy released allows complex I to pump four protons across the membrane.
4 protons just from complex 1. Okay, what about FADH? Does it use complex I too?
No, FADH takes a different route. It delivers its electrons to complex 2, which remember is succinate dehydrogenase from the TCA cycle.
Ah, right, the one embedded in the membrane.
Exactly. FADH transfers its electrons within complex 2, also via iron-sulfur clusters. These electrons are then passed to a mobile carrier called coenzyme Q. But here's the critical difference. The energy drop from FADH's electrons at complex 2 isn't large enough to pump protons, so complex 2 does not contribute to the proton gradient directly.
Okay, 0 protons from complex 2. That seems important for the final count.
It's absolutely crucial for understanding the ATP yield difference between NADH and FADH.
So electrons from both complex I and complex 2 end up on this mobile carrier, coenzyme Q.
Correct. Coenzyme Q, also known as ubiquinone, is a small lipid-soluble molecule that moves within the membrane. It collects electrons from I and 2 and shuttles them over to complex 3.
Complex 3, does it pump protons?
It does. Complex 3, or the cytochrome B-Sea complex, is another major proton pump. As electrons pass through its components, including cytochromes with heme groups, it pumps another 4 protons into the inner membrane space.
Okay, so we're up to 8 protons potentially from NADH, 4 plus 4, but only 4 from FADH, 0 plus 4 so far.
Precisely. Then, complex 3 passes the electrons one at a time to another mobile carrier, a small protein called cytochrome C,
And cytochrome C takes them to the final complex.
to the end of the line. Complex 4, also known as cytochrome C oxidase.
And this is where oxygen finally comes into play, the whole reason we breathe.
This is it. Complex 4 accepts the electrons from cytochrome C. These electrons are now at a much lower energy state. Complex 4 uses them, along with protons from the matrix, to reduce molecular oxygen into water, H0. This final transfer is highly favorable, pulling the electrons down the entire chain. And in the process, complex 4 pumps the last batch of protons, 2 protons per pair of electrons.
2 final protons, so if we add it all up.
For NADH entering at complex 1, it goes through I, 3, and 4. The total protons pumped are 4 plus 2 equals 10 protons per NADH.
Okay.
For FADH entering at complex 2, it bypasses I, goes through 3 and 4. The total protons pumped are 0 plus 4 plus 2 equals 6 protons per FADH.
10 versus 6. That difference directly explains why NADH yields more ATP than FADH down the line.
Exactly. The more protons you pump, the more energy you store in the gradient.
And that gradient, the high concentration of H in the intermembrane space, that's the proton motive force, or delta.
Yes. It's a combination of a chemical gradient, difference in H concentration, so pH, and an electrical gradient, difference in charge across the membrane. It's stored potential energy, like water behind a dam.
Ready to flow back down. And the structure that harnesses that flow is
The grand finale machine. Complex V, better known as ATP synthase. This thing is just incredible. It's literally a molecular motor, a rotary engine powered by proton flow.
a tiny spinning motor making our energy currency.
That's exactly what it is. It has a channel part, Fa, embedded in the membrane. Protons, driven by that gradient, rush back into the matrix through this F channel. That flow of protons causes a central stalk connected to Fa to physically rotate. This rotation changes the shape of the FO part, which sticks out into the matrix. These shape changes in Fa force ADP and inorganic phosphate together, synthesizing ATP.
Wow, so the proton flow directly drives the synthesis. How many protons does it take to make 1 ATP?
The exact stoichiometry is still debated a bit, but the generally accepted number is around 4 protons flowing back through ATP synthase are needed to generate 1 molecule of ATP. Some of that proton movement is also needed to transport phosphate into the matrix.
Okay, so if NADH pumps 10 protons, and it takes about 4 protons per ATP,
10 divided by 4 gives you about 2.5 ATP per NADH.
and FADH here pumps 6 protons.
6 divided by 4 gives you about 1.5 ATP per FADH.
Right, these aren't nice whole numbers, which is why we often see ranges.
Precisely. And if you multiply these yields by the number of NADH and FADH generated earlier, remembering the 2 NADH from glycolysis need shuttling, which we'll discuss, you arrive at that total yield of roughly 26 to 28 ATP just from oxidative phosphorylation alone.
Adding that to the 2 net ATP from glycolysis and the 2 GTP ATP from the TCA cycle gets us right into that 30-32 ATP per glucose range.
That's the calculation. The vast majority comes from this intricate process of electron transport coupled to proton pumping and ATP synthesis via that amazing rotary motor. The efficiency comes from releasing the energy in small, manageable steps instead of one big explosion.
Okay, we've seen the main pathway, the oxygen-dependent powerhouse. But life isn't always optimal. What happens when a cell runs out of oxygen, like during intense exercise, or worse, ischemia?
Yeah, that's when the emergency backup system kicks in, fermentation. The fundamental problem without oxygen is that complex V, the final electron acceptor, gets blocked.
Because there's no oxygen to take the electron.
Exactly. So the whole electron transport chain backs up like a traffic jam. Electrons can't flow. And crucially, this means that NADH and FADH can't unload their electrons and get regenerated back into NAD and FAD.
And NAD, wait, that's needed for glycolysis, right? Way back in step 6.
You can do
You got
it.
Glyceraldehyde
today to
3-phosphate
make tomorrow a
dehydrogenase
better
requires
day.
NAD to
Okay.
function. If the cell
You
runs
can
out of
do
NAD
it.
because the
Let's
ETC
work
is stalled,
today to make
then
tomorrow
even
a better
glycolysis,
day.
which doesn't need oxygen directly, will grind
You
to
can
a halt.
do it.
No NAD, no glycolysis, no ATP at all.
So fermentation isn't really about making more energy. It's about keeping glycolysis running.
Precisely. Fermentation's primary goal is simply to regenerate NAD from NADH so that glycolysis can continue producing that small net yield of 2 ATP per glucose. It's a survival strategy, buying the cell time.
Okay, so how does it regenerate NAD? What happens in our muscles, for example?
In human muscle cells under anaerobic conditions, like intense sprinting, we use lactic acid fermentation. The end product of glycolysis, pyruvate, acts as the electron acceptor. An enzyme called lactate dehydrogenase takes the electrons from NADH and transfers them to pyruvate, converting pyruvate into lactate. And in the process, NADH becomes NAD again.
Ah, so NADH dumps its electrons onto pyruvate, freeing up NADO for glycolysis.
That's the trick. Glycolysis can keep going, making those 2 ATP.
But you build up lactate, which contributes to muscle fatigue and soreness.
It does contribute, yes. And that lactate doesn't just stay there. It gets transported out of the muscle into the blood and travels to the liver. The liver can then take that lactate and, using energy, convert it back into pyruvate or even glucose through a process called the Cori cycle. It's a way of recycling the lactate when oxygen becomes available again.
Interesting cooperation between muscle and liver. And you mentioned red blood cells earlier. They rely only on this.
Completely. Red blood cells have no mitochondria at all, so even floating in oxygen-rich blood, their only source of ATP for their entire lifespan is anaerobic glycolysis, followed by lactic acid fermentation.
Wow, okay. And yeast does it differently, the alcohol fermentation.
Same principle, different end product. Yeast and some other microorganisms want to regenerate NaGO2. They take pyruvate and first decarboxylate it, remove a CO to form acetaldehyde. Then, alcohol dehydrogenase uses NADH to reduce acetaldehyde to ethanol, drinking alcohol. Again, the key outcome is regenerating NADO so glycolysis can continue.
Makes sense. Survival by regenerating NADO. Now let's revisit that 30 versus 32 ATP number. You said it depends on mitochondrial shuttles. Why do we need shuttles?
It's a location issue. Remember those 2 NADH molecules generated by glycolysis out in the cytosol?
Yeah.
Well, NADH itself cannot cross the inner mitochondrial membrane. That membrane is very impermeable. So those electrons carried by cytosolic NADH can't directly reach complex I of the ETC inside the matrix.
So the cell needs a way to sneak the electrons, or at least their energy potential, across the membrane without moving the NADH itself.
Exactly. It uses shuttle systems. These involve pairs of molecules that can cross the membrane, carrying the reducing power of the electrons from cytosolic NADH into the mitochondrial matrix. There are 2 main shuttles used, and they differ in efficiency.
Okay, what's the first one? The more efficient one?
That's the malate aspartate shuttle. or MAS. It's predominantly used in tissues that need high sustained energy, like the heart, liver, and kidneys. It's a bit complex, involving the molecules malate and aspartate cycling across the membrane. But the key outcome is that the electrons from cytosolic NADH end up being transferred to NAD inside the mitochondrial matrix, creating mitochondrial NADH.
So the electrons effectively land on NADA inside.
Yes. And because they become mitochondrial NADH, they enter the ETC at complex I, get the full 10-proton pump treatment, and yield about 2.5 ATP each.
So the MES allows the cytosolic NADH to get the maximum possible energy yield. This leads to the higher total ATP count, the 32 ATP.
Correct. When the MAS is operating, you get the full yield from those glycolytic NADH.
So what's the alternative shuttle? The less efficient one?
That's the glycerol 3-phosphate shuttle, or G3PS. This one is more common in skeletal muscle and the brain. It's simpler and maybe faster, but less energy efficient.
How does it work?
In the G3PS, electrons from cytosolic NADH are first transferred to dihydroxyacetone phosphate, DHAP, to make glycerol 3-phosphate. This glycerol 3-phosphate interacts with an enzyme embedded in the outer face of the inner mitochondrial membrane. This enzyme contains FAD. It takes the electrons from glycerol 3-phosphate and transfers them directly to its FAD, creating FADH within the membrane enzyme itself.
So the electrons from cytosolic NADH end up on FADH inside the membrane.
Yes. And that FADH then feeds its electrons directly into the ETC via coenzyme Q, effectively entering at the level of complex 2.
Bypassing complex 1.
Bypassing complex 1, which means those electrons only contribute to pumping 6 protons, not 10, so each cytosolic NADH using the G3PS only yields about 1.5 ATP instead of 2.5.
So using the G3PS costs the cell about 1 ATP per cytosolic NADH compared to the NAS.
That's the trade-off. You lose about 2 ATP total per glucose, since there are 2 cytosolic NADH. So tissues relying primarily on the G3PS, like muscle and brain, tend to have a maximum aerobic yield closer to 30 ATP per glucose, not 32.
So the type of shuttle used by a particular tissue explains that long-standing variability in the textbook ATP number. Fascinating.
It really does. It depends on the tissue's specific needs, maximum efficiency versus perhaps speed or simplicity.
Okay, we've traced the whole pathway now, and we kept hitting these key control points. Hexokinase at the start, the big one, PFK1 in glycolysis, PDH linking the pathways, and isocitrate dehydrogenase in the TCA cycle. This really emphasizes how tightly regulated this whole process is.
It's absolutely layered with control. The cell is constantly balancing energy production, catabolism, with building things and immobilism. You've got hexokinase, inhibited by its own product, glucose 6-phosphate. That's immediate feedback. Then PFK1, the main commitment step, exquisitely sensitive to the ATP-AMP ratio, citrate levels, really reading the cell's overall status. Then PDH, the gatekeeper to the mitochondria, shut down by its products ATP, acetyl-CoA, NADH. And finally, isocitrate dehydrogenase, controlling the pace of the TCA cycle itself, again sensing ATP and NADH levels. If energy is high, all these brakes get applied, and the cell might shift towards storing glucose as glycogen or converting intermediates into fat.
But sometimes cells seem to choose inefficiency deliberately, which brings us to the Warburg effect in cancer. Otto Warburg noticed that tumor cells often rely heavily on glycolysis, even when there's plenty of oxygen around. They ferment glucose to lactate instead of sending pyruvate to the mitochondria. Why would they do that? It seems so wasteful energetically.
It seems counterintuitive if you only think about ATP yield, yes. But Warburg's insight and subsequent research suggest that cancer cells prioritize rapid growth and proliferation over maximum energy efficiency.
So it's not about the energy, it's about the building blocks.
Largely, yes. Running glycolysis really fast, even if incompletely, allows the cell to siphon off intermediates from the pathway to use for biosynthesis. For example, they need lots of glucose 6-phosphate and fructose 6-phosphate to feed into the pentose phosphate pathway, or PPP.
What does this PPP make?
The PPP generates 2 crucial things for dividing cells: NADPH, which is needed for synthesizing fatty acids and reducing oxidative stress, and ribose 5-phosphate, which is the sugar backbone needed to build nucleotides, the A, T, C, and G of DNA and RNA. You can't replicate your DNA without it.
So fast glycolysis fuels DNA synthesis.
Exactly. Also, intermediates like citrate can be exported from the mitochondria, even if the TCA cycle isn't running at full speed, to provide acetyl-CoA in the cytosol, which is the precursor for making new fatty acids and cholesterol for membranes. Rapidly dividing cells need lots of new membranes, so aerobic glycolysis, the Warburg effect, is likely a metabolic adaptation that supports rapid biomass accumulation, even at the cost of ATP efficiency.
That's a really different way of looking at metabolism, not just energy, but resource allocation for specific goals like growth.
Yeah.
Now let's flip to when the system breaks down involuntarily. Metabolic crisis, like ischemia, lack of blood flow, meaning lack of oxygen.
The consequences are swift and devastating. No oxygen means complex. Complex 4 stops. ETC backs up instantly. ATP synthesis via oxidative phosphorylation plummets.
And the cell runs out of energy fast.
Very fast. And when ATP levels fall critically low, the cell can't power its essential ion pumps. The most crucial one is the NAHCO ATPase, the sodium-potassium pump, which uses a huge amount of ATP to maintain ion gradients across the cell membrane.
What happens when that pump fails?
Sodium ions, which are normally pumped out, rush into the cell following their concentration gradient. Water follows the sodium osmotically, the cell swells up, the membrane can rupture, and you get irreversible cell injury and tissue death. It's a major mechanism of damage in stroke and heart attack.
And certain poisons work by directly mimicking that oxygen deprivation, by blocking the ETC itself.
Yes, the classic mitochondrial poisons are terrifyingly effective. Cyanide, carbon monoxide, CO, and azide all specifically target complex 4 cytochrome C oxidase. They bind extremely tightly to the iron atom in the heme group within complex 4, preventing oxygen from binding and accepting electrons. It's like throwing a wrench into the very last step. ATP synthesis halts almost immediately. Fatal.
Are there poisons that hit other complexes?
Oh, yes. Rotenone, which is used as a pesticide, specifically inhibits complex WAR, blocking electron entry from NADH. Antimycin A, an antibiotic, blocks complex 3, stopping electron flow between coenzyme Q and cytochrome C. And then there's oligomycin. It's interesting because it doesn't actually block electron flow down the ETC. Instead, it directly targets complex V, the ATP synthase.
How does it block ATP synthase?
It physically plugs the proton channel, the EFA part. So the ETC can still pump protons, building up the gradient, but the protons can't flow back through ATP synthase to make ATP. The motor is jammed.
So the energy just dissipates.
The gradient builds up until it's so strong it actually inhibits further proton pumping by the ETC due to back pressure, but no ATP is made.
It really shows how these complexes are specific targets, which also means they can be targets for medicines, right? Like metformin for diabetes.
Metformin is a fascinating case. It's one of the most widely prescribed drugs in the world. Its exact mechanisms are still debated, but a primary effect seems to be mild inhibition of complex I in the mitochondria, particularly in the liver. Mild inhibition, so it doesn't shut things down completely. No, it just dials it back a bit.
Yeah.
This slight decrease in ETC efficiency leads to a small drop in ATP levels and an increase in AMP levels within liver cells. This change in the energy state, specifically the increased AMP-TP ratio, activates an enzyme called AMPK. Activated AMPK then signals the liver cell to reduce its production of glucose, gluconeogenesis. Since excess glucose production by the liver is a major problem in type 2 diabetes, metformin's effect on complex I helps lower blood sugar.
Amazing that such a subtle tweak to mitochondrial function can have such a big therapeutic effect. And then there are the uncouplers. These deliberately make the process inefficient.
Yes, uncouplers break the link. They uncouple electron transport from ATP synthesis. They essentially provide a leak pathway for protons to flow back into the matrix, bypassing ATP synthase altogether. The dangerous diet drug DNP, 2-frictyl-4-dinitrophenol, did this. Protons flow back, the energy stored in the gradient is released, but since it doesn't go through ATP synthase's motor, it's just dissipated as heat.
So the ETC keeps running like crazy trying to maintain the gradient, burning fuel, but generating heat instead of ATP.
Exactly. That's why DNP caused rapid weight loss, but also dangerous overheating, hyperthermia. However, our bodies actually use controlled uncoupling naturally. Brown adipose tissue, or brown fat, is rich in mitochondria, containing a specific uncoupling protein called thermogenin, or UCP1. When activated, like in response to cold, thermogenin allows protons to leak back across the membrane, generating heat instead of ATP, which helps keep babies and hibernating animals warm.
So controlled inefficiency for heat generation. Very clever. Lastly, the long-term health of these mitochondria. Failures seem linked to chronic diseases. especially neurodegeneration.
There's a strong connection being researched. While the ETC is usually very efficient, a small percentage of electrons can leak out prematurely, especially from complex I and III, and react directly with oxygen to form reactive oxygen species, ROS, like superoxide radicals.
Rats.
Yes. The cell has antioxidant systems to deal with normal levels of ROS. But if mitochondria become damaged or dysfunctional, due perhaps to age, toxins, or genetic factors, ROS production can increase significantly. This excess oxidative stress can damage cellular components, proteins, lipids, DNA. There's growing evidence implicating mitochondrial dysfunction and ROS damage in the progression of diseases like Parkinson's, Alzheimer's, and ALS, suggesting that maintaining the health of this energy-producing machinery is critical for long-term health, especially in high-energy demand tissues like the brain. Hashtag outro.
Well, that was quite the journey through the cell's engine room. We went from that initial small ATP investment in glycolysis, followed the carbons through pyruvate oxidation and the TCA cycle, harvesting all those NADH and FADH arrows.
Right, loading up the energy carriers.
And finally saw how oxidative phosphorylation caches them in on the inner membrane for that massive ATP payout, around 26 to 28 ATP just from that stage alone.
All driven by that elegant stepwise release of energy through redox reactions and the creation of that proton gradient, Peter Mitchell's chemiosmotic theory in action.
And we finally cleared up that 30 versus 32 ATP mystery. It comes down to those mitochondrial shuttles, the efficient MAS versus the less efficient G3PS, determining how much energy we get from the NADH made on the cytosol.
Exactly. And it highlights the central role of acetyl-CoA as this hub, where pathways for carbs, fats, and even proteins can all converge to feed into the energy production pipeline.
We really stand on the shoulders of giants here. Krebs mapping the cycle, Mitchell figuring out chemiosmosis.